Good day and a very welcome to assessment of the fetal status using the cardiotachograph. And today we're going to do lesson 4, which the first part, both parts is the physiology of the fetal heart rate control. The first part will be on the uteroplacental exchange and the autonomic and the somatic components of the central nervous system. And the second part will be on the baroreceptors and the chemoreceptors, the adrenal glands, and some other factors that also influences the fetal heart. Now, I'm always saying that it's so important to understand the physiology of whatever you work with, because if you do understand the physiology, it's so much easier to do the correct assessments, after you've done the assessments, to interpret them correctly, and then also to decide on the correct management of the problems that you have identified. So I hope you enjoy it, and I hope it makes a lot of sense to you. Now, what you see here in front of you is a cut through the placenta, and I will use that to explain to you the uteroplacental exchange. The uteroplacental exchange plays a very big role in the oxygen exchange between the mother and the fetus during pregnancy as well as during labor, and specifically during labor there are some very important physiological aspects that happen. So this is extremely important, and later on when we start with the interpretation of the CTG graphs, we will come back to this again, especially when we come to hypoxia and deceleration, specifically during labor. Now, as I've said, you have a cut through of the placenta here in front of you. Here is the liquor and the uterus, and the baby will be here somewhere. There's the umbilical cord going to the baby, and as I've said, this is enlarged part of the placenta. Now, in the very early fetal development, when the fertilized egg has implanted here in the wall of the uterus, it sends out finger-like protrusions. And these finger-like protrusions develop, and in them develop a blood circulation system. And as they develop and as they grow through the uterine wall, they literally grow through the maternal blood vessels as well. And that causes these villi to be absolutely surrounded and bathed in maternal blood. So here you can see the villi, these finger-like protrusions, and in each one of them there's a small fetal artery and a small fetal vein that branches and come together eventually and form the umbilical vein and the umbilical artery. Here, this part of the placenta, this part of the photo here is the uterine wall, the muscle of the uterine wall, and here you can see maternal arteries coming from the mother, and then it has been, as I've said, literally these finger-like protrusions grow right through them so that they rupture, and the villi is being surrounded by maternal blood. Now, the oxygenated blood comes from the mother in the uterine arteries. The villi is surrounded by maternal blood. So oxygen in the maternal blood diffuses then from the maternal blood through these villi into the umbilical veins. In the case of the fetus, here in the placenta, the veins transport the oxygen-rich blood. These veins all get together and form the umbilical vein, and with the umbilical vein, oxygenated blood goes to the mother. Same way, CO2 and offal-rich blood come from the umbilical artery, branch here in the smaller umbilical arteries, and then the same way, CO2 and all the offal products are going into the maternal blood and is eventually taken away by the maternal veins. Now, if we look at this uteroplacental exchange specifically during labor, there are some very important aspects to take into consideration. And we will come back again and again to it again when we interpret the fetal heart, and there are some decelerations and fetal compromise, etc. We will refer to this uteroplacental exchange during labor again and again. So it's very important. Now, what happens here is if this is the placental, the uterine wall, as we've said, the muscle of the uterine wall, and these are the uterine arteries coming in here. The uterine arteries are spiral formed. They have lots of kinks in them, which makes them very easily compressible. So the moment that this uterus contracts and this muscle contracts, it literally occludes all these maternal arteries. So during the duration of a contraction, no new maternal blood comes through these arteries into the intervallar space. Now, important here is that although these arteries are being occluded, this villi are still being surrounded by maternal blood. So there's still oxygen in the maternal blood. If you visualize yourself in a room and all the oxygen inlets are being cut off, like in the windows are all closed, etc., there's still some oxygen for you to live on and survive for quite a while, and this is the same case here with the fetus. And then also the other thing is, in the case of healthy fetuses, they normally have enough reserves to survive this period where no new oxygen is going to the placental blood. Now, after contraction, when the uterus is now relaxing, all these the muscle is relaxing and all these arteries are opening again, and new fresh oxygenation or new oxygen blood full of oxygen can go through. Now, except for the fact that there is that the fetus have reserves and that there's blood in here, the other very important thing that helps the fetus to survive is the fetal hemoglobin. Now, we all know that on the hemoglobin sits on the red blood cells, and oxygen is being transported in the blood on the hemoglobin. Now, fetal hemoglobin differs a little bit from normal, our mature hemoglobin, in the sense that it has a much higher affinity for oxygen than normal mature hemoglobin. So it can take oxygen from this blood here in the villi at a much lower PO2 than what our normal mature hemoglobins can do, and that also helps this fetus to survive in the case where there's not enough oxygen initially. The problem comes when this labor with the cut off of oxygen over periods where there are contractions last for a long time. So when we have prolonged labor, these reserves might become exhausted, and there's not enough oxygen over a very long period for this fetus to survive. That's the one thing. And then the same is also when there was placental insufficiency even before labor started. The placenta is not normal, the fetus already has been disadvantaged and doesn't have these reserves to cope with and to survive this periods of less oxygen. And also when the contractions are very long and when the frequency of the contractions are very high. When the frequency of the contractions are very high, the rest periods between the contractions are so short that the arteries barely has opened before the contraction starts again, and they are being occluded again. So these factors play a very important role, and as I've said, we will come back to them again. Then if we look further at the physiology of the fetal heart control, we talk about the central nervous system and the control of how do the central nervous system control the fetal heart. And we all know that the central nervous system have an autonomic and a somatic component. The autonomic component consists of sympathetic and parasympathetic systems, And the parasympathetic system is responsible for the decrease of the fetal heart, and the sympathetic system is responsible for the increase of the fetal heart. And these facts, the influence of the sympathetic and the parasympathetic system and the increase and the decrease in the fetal heart, you will hear many more times in this lesson, in the next lesson where we start interpreting it, and specifically in the lesson 6 where we do the abnormal CTGs. So this is something that you have to store in your brain. Sympathetic increases the fetal heart rate, parasympathetic decreases the fetal heart rate, and there's a constant interaction between the two. Now, if we look further at this autonomic and sympathetic and parasympathetic systems, there's a interaction between these parasympathetic and sympathetic nervous system all the time. And that cause an increase and a decrease in the fetal heart rate all the time. As I've said, sympathetic increase, parasympathetic decrease, and as they interact with the fetal heart and the fetal heart rate, it causes an increase and a decrease in the fetal heart rate all the time. And that is what we eventually call baseline variability. That means the fetal heart rate is never constant. And for this sympathetic and parasympathetic nervous system to interact with each other in a healthy way, we need enough oxygen. So when there's a good baseline variability, it tells us that there's good enough or well enough oxygenation to the sympathetic and the parasympathetic nervous system. Now, if we look at the central nervous system, we'll specifically look at the somatic part of the central nervous system, which is responsible for body movements. And we all know that the moment when we move and our bodies move and we start exercising, our heart rate increases. Now, it's exactly that, and even more so in the case of the fetus. The somatic component of the central nervous system makes the fetus to move. It causes body movements. And the moment that the body moves or the fetus moves, it causes an acceleration of his heart, of the baby's heart. And that, the same way as the parasympathetic and the non-parasympathetic nervous system, tells us that if there are accelerations together with fetal heart rate, together with fetal movements, that the somatic nervous system gets enough oxygen and glucose so that the fetus can move and that the fetal heart rate accelerates with that. So accelerations are usually a very good thing, and we love to see accelerations. For some or other reason, during labor, sometimes accelerations disappear, and we're not 100% sure what the reason for it is. In some cases, there are still accelerations, and in some cases, it disappears without being abnormal. The fact that they're not there doesn't make the fetal heart pattern in any way abnormal. Then just one other thing regarding and this is coming again to the parasympathetic system, to a large extent, parasympathetic and sympathetic, but also the somatic nervous system, and that is that fetuses have periods of sleep, what we call quiescence periods, and periods of activity. Now, and it's been described by a term cycling. It means it happens over periods of time. There are periods of activity and there are periods of quiescence. And like you can see on this graph, in the periods of quiescence, there's very little interaction between the sympathetic and the parasympathetic nervous system. And that causes decrease of baseline variability, as you can see here. Then the fetus wakes up, and then there's fetal movements, there's variability, there are accelerations, etc., and now the fetus is wide awake. So when you see a decreased variability, one always needs to make sure that it's because of fetus, whether the fetus, make sure whether the fetus sleeps or whether it's because of some other factors, like, for example, hypoxia or not. Now, these sleeping periods last more or less 50 minutes. They can be shorter. They're seldomly much longer. So if you have a period of decreased variability, one needs to carry on monitoring the fetal heart rate for a little bit longer to make sure that this is because of a sleep period and not because of hypoxia. And this brings us to the end of lesson 4, part 1, and I really hope that you've learned something new here and that you will really remember what you've learned. You have to make sure that you do remember it for the quiz at the end of part 1, at the end of part 2, but also for when we come to the interpretation of all the other slides.